And I would like to introduce Dr. Fabrice LAURENT. He's an immunologist and has been working with INRA since 1994. And his focus is host pathogen interactions and intestinal immunity. The floor is yours.
So thank you for the introduction, and also thank you, Pierre-André, for this marvelous meeting and for this invitation. In my laboratory, we are actually interested in host pathogen interaction, and we study mucosal immune responses. And basically, we do work with two infectious models. One is affecting the livestock, which is Cryptosporidiosis, and the other one is affecting the poultry industry, that is coccidiosis, we heard about already this morning. What I would like to discuss with you is I would like to state that it's very important to investigate these immune cells, immune effectors, together with the microflora, that they all together impact the outcome of the infection, leading to protection or to physiopathology. So I will use some examples of the work we have done this last year in the lab to show you how we can illustrate this. But before I go with this infectious model, I just want to state that homeostasis is at birth when we have the first incoming antigens coming from the flora, of course, but also from the food. And these gave the very first signals to epithelial cells and residing in immune cells. And these, what we can call the physiological inflammation, we have some answer, production of mucus, of antimicrobial peptides, and some IgA. But this is quickly, tightly regulated, and there's no what we call some kind of inflammation. But when we get an infection, some kind of enteric infection, of course, we get an inflammatory response from these epithelial cells, but also from the immune cells that are coming into the mucosa. And if it works as you expected, you had a quick protection, and then you go back as soon as possible to homeostasis. But if you have unregulated immune response, then you got some pathological inflammation, and you got a pathology with intestinal lesions. Okay, I will jump now on the first model, that is Cryptosporidiosis. As you can see over here, you have this extracellular stage of a parasite that will jump from epithelial cells to replicate. Few words about Cryptosporidium, because I know many of the main people here are interested in poultry, maybe you don't know the parasite. So it's a single cell parasite that just grows on top of epithelial cells, and it makes its whole cycle in 3 to 4 days. It's really restricted to the apex of epithelial cells, and there's a high prevalence in the livestock, and it's also a zoonotic disease. It affects young cows and young children, and also immunodeficient individuals, because the severity of the infection is really linked to the immune status of the animals. So it's a beautiful model to study intestinal immunity, especially in neonates. And these last days we tried to decipher all these mechanisms that are involved in protection or physiopathology. Just to show you how it looks like intestinal villi. This is a normal one, and then you see all the parasite all along, and actually all epithelial cells can be infected. So of course there's a dramatic change in the regulation of these cells. So what happened when the parasite first encountered the epithelial cells? Epithelial cells just give a first answer. They just produce small amount of antimicrobial peptides, and they can undergo apoptosis. They are inducing their own cell deaths. But usually this is not enough to kill the parasite, especially because the parasite has developed evasive mechanisms to most of these different mechanisms of killing. So, but also epithelial cells, they produce small molecules that are called chemokines, and these chemokines attract immune cells. In this case, these immune cells will control the infection. But we tried this last year to understand which of these cells were important in the protection. That's why, for that, we used mouse model, because we can use a lot of different transgenic or knockout mice to try to decipher the complete mechanism. The first set of experiments that we did was to look at use mice that are deficient for T or B cells. In this case, this is a curve of where we see the excretion of a parasite. There's the acute phase of the infection, and then the animals are protected. And when you don't have these B or T cells, you also see the exact same curve, actually. Adaptive immunity is only important to completely clear the infection, but it's not important to control this acute phase of infection that is leading to this diarrhea in these young animals. So we knew from that was innate immunity was key, but which cells? And we first started to look at molecular phagocytes. It's a large family that you can find some macrophages or dendritic cells, and we wanted to know which were important, if these cells were important, and which one. So the first model that we used was to use some CD11, CD2 mice, whatever the name is for you. Just remember that with that model, we can have a transient depletion of these all mononuclear phagocytes, all this family, dendritic cells, monocyte, and macrophages. They're all depleted, sometimes almost 100% for dendritic cells, and for this one, almost 60% of the cells are depleted. And with this model, you can clearly see that the curves after depletion, you get a much higher level of parasite in the intestine of these mice, of these neonate mice. But then, as it's just a transient depletion with a diphtheria toxin, the new cells coming from the bone marrow just come back, and then you regenerate all these subsets, and then the neonatal mice can control the infection. So for sure now, we knew that at least dendritic cells or monocyte and macrophage were clearly important to control this acute phase of infection. And we continue to work with these different models. And to differentiate if there was a macrophage or dendritic cells, we first focus on dendritic cells. And we use different cytokines where you can amplify some subsets, for example, and you inoculate FLAG3 ligand administration, so you just amplify this different subset. By doing this, we were increasing the resistance to infection. So it was a good prognostic that these cells were very important. And very lately, we just published a paper a month ago. And for that, we use battery deficient mice. These mice have deficiency for the development of some of these subsets of dendritic cells. And clearly, these CD103 dendritic cells are really at present at very, very low level. And these mice clearly are much more infected than the conventional one. And if you just even look at these young animals, 5 weeks later, you can still see the parasite in the intestinal villi. So with all these different models, now we knew that these dendritic cells were really key for controlling the infection. The mechanism by which they do that is probably because they produce large amount of IL-12, and actually the first interferon gamma response, that is a very important cytokine to control the parasite in the epithelial cells. And probably this signal will be amplified by other innate cells, like innate lymphocyte. But at the same time, we have found that there's also a lot of inflammatory monocytes that were recruited at the site of the infection. And to study the function, we just look at, we use another model that is deficient for CCR2, it's a chemokine receptor. And in this case, in these mice, the monocyte cannot get out of the bone marrow, and they cannot be recruited in the lamina propria. So that was a good model to study that function. And with these mice, that only very, very few inflammatory monocytes, although these monocytes are present in very high level in normal mice, and they're in close contact to infected epithelial cells. With this model, we were expecting that the mice will be much more infected, like we have seen previously with depletion of dendritic cells, and it was not the case at all. So it was a bit surprising for us. We say how this 13-fold increase in inflammatory monocyte do nothing. That was a bit surprising. So we we start thinking maybe they're not involved in protection, but maybe they're involved in some deleterious effect on the mucosa. So, as we know that Cryptosporidium is inducing diarrhea, and we know that they are also modifying the transepithelial resistance, we have done experiment, and in vitro it's clear. You can see with the Stranwell chambers, you infect with crypto and you see a drop in the transepithelial resistance. We have done this also in vivo. Well, we'd use a different mechanism. We'd use the term feedchannel, and then normally at homeostasis there's only few of the feedchannel that you give by gavage that can go through the epithelial cells and go through the blood, and you can measure the fluorescence in the blood. But when you do that with infection with crypto, you clearly see an increase, and it's depending of the virulence of the strain of crypto you are using. So knowing all this, we also made some experiment with a postdoc, and he was able to make a long story short that Cryptosporidium by itself on epithelial cells was also affecting some of the adherence junction, and in case E-catenin and beta-catenin. And in vivo, we found that this mechanism is probably due to the very large amount of IL-1 alpha and TNF alpha, that IL-1 beta and TNF alpha that is produced by these inflammatory monocytes. And as it was mentioned this morning, these two cytokines are able to destroy the tight junction, and then for explaining why we have this increase in permeability. So to summarize on the very first part, so just imagine for this model, you got Cryptosporidium coming into epithelial cells, and the chemokines attract these immune cells in the lamina propria. Some of these cells are more involved, inflammatory monocytes, and more involved in the increase of permeability, and we know that there's acute diarrhea in young cows, so maybe that's one of the mechanisms. And some other cells, by chance, are actually protecting the epithelium, like this different subset of dendritic cells, by producing interferon gamma. And there's a lot of different intracellular mechanisms that can block the parasite development in response to interferon gamma. If you want to have some a review, we made that a review last year, so if you want to have some more information, please go and read it. Okay, so how can you use this this information to see how we can strengthen these mucosa in neonates? So we knew, probably everyone's know now in the audience now, what are the toll-like receptors. So innate receptors that are present on the surface of a cell or endosomal compartment. And when you bind some of the different ligands coming from bacteria or viruses, parasites, and fungi, then you induce some inflammatory signals in the cells. And by chance now they have also new synthetic ligands that are available commercially, so you can buy and try this. So we have done, it's quite a long different experiment with different toll-like receptor agonists. And we have shown that when we just inoculate these agonists to the newborn mice, we were quickly protecting from against Cryptosporidium. And this mechanism, sometimes you can have a 90% protection in less than 24 hours. So it was very efficient. But what I would like to share with you is like this, with different models, we got different type of response. What was interesting is like for CpG ODN, like mimic bacterial DNA, for example, we got a protection, and poly-IC that mimics some nucleic acid from viruses, you got also a protection. When you do an antibiotic treatment with a broad spectrum of antibiotics, removing almost all bacteria, then you lost the protection with that, with poly-IC, and you don't lose it with CpG ODN. And it took a while for us to understand how this was working, but in the case of poly-IC, there's some signals coming from flagellate bacteria that binds to toll-like receptor 5 that is present on the surface of these dendritic cells, and that induce some MID88 signaling, so a different set of cytokine expression. And that, together with the first signal, gives protection. But if you remove that signal, this pathway only is not protective. So, and this is not the case for CPG because this goes for strong activation of MID-ADH pathway. So now, of course, this was just a proof of concept, but we are just seeking for a natural compound that can just make the same effect. I will now jump on the second model, so coccidiosis. It's just a picture of sexual stages, flagellate stage at a male that will try to find a female to make an oocyst. Sorry. Okay. We heard about coccidiosis this morning, but just to say a few words about the disease. It's ranked in the top 20 vet disease, and it's the most prevalent disease affecting the broiler industry. There's a strong economic impact. It's $3 billion per year, and due to production losses and treatments. We do have a vaccine for layers, but it's a bit costly, and we have chemotherapy also for broilers that is mandatory because the disease is very prevalent. But there's a lot of resistance to all these molecules. So the clinical sign of coccidiosis is decreased growth rate, weight losses, mortality, and decreased egg production. But today I would like to focus on one of the strains that infect one particular place of the intestinal tract of chicken. It's like Emeria tenella that causes hemorrhagic cecal coccidiosis in young poultry. And we wanted to understand what was the mechanism for this hemorrhagic cecal lesions. For that, we have developed several transgenic lines for Emeria that we can have expressed fluorescent, and we now do have also a unique tool for screening of anti-Emeria compounds in the lab. So the working hypothesis was like, in normal conditions, then Emeria infect epithelial cells, start to develop. Some of the chysons go back in the lamina propria, and then they release some free stages. And probably we expect some strong inflammation because it's within the lamina propria, probably inflammatory monocyte, maybe like we have seen with a Cryptosporidium model. And this will increase intestinal permeability, and maybe translocation of bacteria from the commensal flora will increase this inflammation, leading to this huge lesions that we can see with this infectious model. So we first look at the macrophages as, and you can see here we have the green parasite, we have green chysons. You got macrophages that comes all around these chysons. So we do know that these cells are recruited during the infection around infected sites. And also that these macrophages, when you isolate them, they produce iNOS, which is an enzyme that will produce NO and superoxides. So in these cases, these agents can cause lesions, but we still have to demonstrate this. So to see the importance of microflora, we use axinic chickens. We're actually producing roast chickens, so broilers, without any flora, and then we infect it with Emeria tenella and look at the lesions. That was very interesting to see that compared to normal animals, the axinic ones have almost no lesion. And even if you increase the numbers of the oocysts as an inoculum 10 times, you see there's no lesion on the cecal coccidia. So that was very exciting for us, and we say maybe the bacterial flora is making it all. But the things are probably much more complex, because if you look at the numbers of a parasite that replicate in these axinic animal, it's much lower too. So the next challenge for us now to understand what is the mechanism. We should determine what is due to parasite replication for the lesions and why it is going like this. Or it is due to the bacterial flora, that in maybe some breaches in the mucosa, and some bacteria can come inside the lamina propria, or maybe both. So what we do also, it's just an ongoing project that is called Integrity Project. We do work with also from our colleagues that also work in Tours, but they are nutritionists, and I am not, so I'm not going to go into much details. But what they want to do is to modify the alimentation of young birds with insoluble fibers in the starter feed for the first days. And we will see if by changing the microflora, we do impact the lesion score at the end. Something I would like to share with you also is that good and bad guys exist in immunology, and by guys I mean immune cells or effectors. And you will see that virtually all cells can be good and bad at some times depending on the condition. I will just illustrate with this with another infection by protozoan parasite, that is Toxoplasma gondii. And if you look at the literature, you can see that, for example, if you infect orally mice, C57 mice with Toxoplasma gondii, you can produce a huge interferon gamma production by CD4 T cells. And this huge interferon gamma production in the lamella propria will induce ileitis and mouse death. So if you just look at these data, you will just conclude that interferon gamma appears deleterious for controlling the disease. But if you go back in the literature and you see how many papers are published showing that, for example, with attenuated strain of Toxoplasma gondii, if you use mice that do not produce interferon gamma, they will die quickly. And if you look at how the mechanism is, usually all this interferon gamma produced by various immune cells, they control the parasite especially into macrophages, but in different cells. So for this, you can see that every molecule, depending on the condition, could be beneficial or detrimental for immune responses. So this depends on the pathogen virulence, the host genetic, but also different what I call environmental factors, but the age of the animals, if there's co-infections or not, presence of the flora, the food, and so on. So the goal is to induce or slow down immune mechanism very quickly, depending on the modeling condition, to clear the infection and go back to homeostasis as soon as possible. So the take-home message from my talk is that innate immunity is important, especially in neonates and young animals, for controlling pathogens in intestinal epithelial cells. I've shown you that we know that dendritic cells are crucial for antigen presentation and orchestration of adaptive immune response. This is in the textbook. But we also showed recently that these cells are major actors of intestinal innate immunity and primary target for immuno-stimulation strategy. Host microbiota composition is not only important for intestine immune development, as been shown by Bernd previously, but also they can modulate host response during infection or immunomodulatory treatments. As I just mentioned, some specific cells or effector can be key for protection, but also be involved in adverse effect and pathology. And I hope I convince you that deciphering all these immune mechanisms is the starting point for developing targeted strategy to strengthen immune response in the gut and, of course, at the end, improve gut health. Just would like to acknowledge all the collaborators in the group that have done the work on Cryptosporidium and Emeria, but also some other collaborators that work in the project for the Integrity Project, and also especially the people in the facilities that help us to develop all these mouse, young cows, and also axenic chicken in the platform of infectiology. Thank you very much.
Thank you, Fabrice. Okay, we have a few questions. So, do you want to pick from the top?
So, how many questions I have to answer?
Yeah,
all?
You have.
I pick up.
You can pick up 2 or 3 questions. It depends on the length of the answers.
Just take the toll-like receptor important to improve immune response? Yes. Are they a good target for us to manipulate diet? I think we can, but we need to know that these toll-like receptors are tightly regulated in the intestinal mucosa. As soon as the microflora come, for example, if I can take, maybe it depends of which toll-like receptor we are talking about. For example, toll-like receptor 4 is quickly regulated just in the very first hours after birth because of the presence of a flora that increase microRNA in epithelial cells. And in these epithelial cells, they become quickly non-responsive to LPS. Of course, this is one of the mechanisms to avoid strong inflammatory response because of the presence of a large amount of LPS in the gut.
Can you select birds more resistant to parasite?
The differences between a bacterial pathogen compared to parasite? It depends what you call parasite. If you're talking about worms, it's completely different. But with intracellular parasite like Cryptosporidium, for example, some of the mechanisms could be similar. And actually, we for immuno-stimulation, it's something that is not yet published, but we can, with new toll-like receptor agonists, you can increase some of the immune response that are not yet present with a natural response, and this mechanism also important for controlling viruses. So probably yes, with intracellular protozoan parasite, they have much in common with intracellular bacteria and intracellular viruses.
Thank you. That one question. First question, can you select birds more resistant to parasite? The last question.
I think there's people have tried that. The Fayumi birds, for example, I think they're more resistant to the parasite, but it's difficult because I think in the broiler industry they don't want to change much the genetics of the animals. So yes, probably, but you need better to select what is really important, and that we need to do a lot of much work, basic knowledge first.